Hello everyone and welcome to Instant Biology by Dr. Nilab. I am here with you again with another very interesting topic, and the title of the topic that we are going to deal today is Cosmid vectors. So you all would be knowing that in our playlist we have already talked about PBR322 and PUC19 vectors, and in the same series today I am going to discuss about the Cosmid vectors. For those of you who understand Hindi better, I have already made a video in Hindi on the same topic. For those of you who understand English better, this video would be completely in English. Now, before starting this particular video, let me just tell you what we would be dealing with in this particular video. We would be talking about the basics of Cosmid vector. I would be discussing about the features that Cosmid vector brings. Apart from that, I would also be dealing with some of the previous year questions. I would be dealing with the importance of the cos site and terminase enzyme also. So let us start about discussing about the Cosmid vectors. So first thing first, what is a Cosmid vector basically and how it is prepared? So you should understand that Cosmid vector is just like a plasmid vector only, but it has some of the additional features. Now, as I've written over here also, you can see on your screen that Cosmid vectors are nothing but they are actually plasmids indeed. They are actually plasmids. It's just that they are having one additional feature, and that feature is that they comprise of 250 base pair of DNA, and this DNA is coming from the lambda phage. This is an important point that you should remember. So 250 base pair of lambda phage DNA is being used over here, and the rest of the thing is coming from the plasmid itself. So this 250 base pair of lambda DNA is the minimum amount of DNA that is present inside a Cosmid vector. Now, what are the features or what is the basic composition of this 250 base pair lambda DNA? That means, what are the characteristic features of this 250 base pair? What is present inside this 250 base pair? So actually the phage lambda genome has an important feature or important site called as cos site. Why am I saying this phage lambda genome? Because this lambda phage DNA, or you can also call it phage lambda DNA, is obtained from the phage lambda. That means it is a bacteriophage virus. That means we are taking a small portion of the bacteriophage lambda and we are inserting it inside our plasmid. For instance, let me just tell you, suppose this is a plasmid. I'm just drawing a very basic diagram over here. So suppose this is a plasmid and this is the lambda phage important region or important site, and I'm calling it as cos site. I'm calling it as cos site. So what we do is, to our plasmid we add this cos site and it becomes something like this, right? So this over here is now our cos site. So the cos site is obtained from the lambda phage DNA or lambda phage genome. Now what does the cos site do, or what are the features of the cos site? We would be talking about that in just a moment. Primarily, cos site gives or provides us sequences yielding cohesive ends. What do you mean by cohesive ends? Cohesive ends means sticky ends. So this cos site provides us sticky ends. When does it provide us sticky ends? When the cos site is cleaved by a restriction enzyme. So in other words, we can say that inside the Cosmid vector, this cos site will be present, and this cos site can be cut off or can be cleaved by a restriction enzyme. Okay, so cos site is present inside the Cosmid vector also, and cos site is also present inside the lambda phage genome. From the lambda phage genome only we are taking the cos site and inserting it inside the Cosmid vector. Up till here you would have understood. Now let us move on to the other point. So why is cos site needed? So cos site is a site, as I have already told you, cos site is a site wherein the enzyme called as terminase. So you have to remember the name of the enzyme. The name of the enzyme is terminase enzyme. So this terminase enzyme will be binding at the cos site and it will be cleaving it. So this is the feature of the enzyme terminase. Now where is this enzyme terminase present? The enzyme terminase is present inside the, or it is coded by the lambda, it is present, or it is coded by the lambda phage genome. Right? So it is coded by the bacteriophage, this terminase enzyme, and this terminase enzyme goes and cleaves off the cos site, thereby creating cohesive ends. Now let us move on to the next point. What are the typical features of a cosmid? What are present, or what are the important constituents of a cosmid vector? The first thing is replication origin. Of course, we know that because the cosmid vector is derived from the plasmid only, so replication origin, or in other words you can call it as origin of replication. Origin of replication. So obviously this origin of replication would be present inside the cosmid vector. Apart from that, unique restriction site should also be present. What do you mean by unique restriction site? Unique restriction site simply means the site from where the restriction enzyme can cut off this particular vector. This is the second important feature. The third important feature is selectable markers. For example, antibiotic resistant markers are present over here in the cosmid vector. Now, through the selectable markers, you can understand, or you can detect that which particular bacteria now has the gene of interest, or now has the vector. You can detect that easily. So talking about that screening and selection, I have already made videos on that. You can check out the playlist and you can get a better understanding about this. So selectable markers also we have discussed. Now cosmid vectors are not present naturally. That means they are artificial vectors. These artificial vectors can be synthesized only by RDT. I will give you a nice example over here. For example, you want to purchase a computer. Now different companies will provide or manufacture different components of a computer. For example, let us say Samsung manufactures a particular component, and the Intel manufactures a particular component, so on and so forth. So now if you want to get a high-end PC, what people do is get the entire PC assembled. So they purchase different components from different companies and they get their PC assembled. So the same thing is being done over here. So the plasmid is basically coming from the bacteria, and the cos site etc. are coming from the lambda phage genome. That is why we are saying that it is constructed through RDT. It is an artificial vector, and through RDT it is constructed. So this is the next important point. Okay, now let me just quickly show you the diagrammatic representation of a cosmid. As I've already said, as I've already told you, that origin of replication, ORI site, is present inside the cosmid vector. Because of this origin of replication only, the cosmid vector can replicate autonomously. That means independently, independent of the host machinery. This particular cosmid vector can replicate on its own. Next point is that it is having the cos site. So you can see this is the cos site, this green thing, this is your cos site. I will be telling you in just a moment that what is the importance of the cos site. Apart from that, antibiotic resistance gene is present over here. This is called as the selectable marker. I just told you. Apart from that, there are restriction sites also. You can see over here there is an ECO R1 site, and there is a SMA1 site also that is present over here. Now let us understand what is the cos site. Now cos site is present inside the lambda phage genome, and it is cleaved by the terminase enzyme. After cleaving by the terminase enzyme, it produces cohesive ends. Now with the help of the cohesive ends, what happens is multiple lambda phage genomes can combine together. That means they can align together in this way. And the cos site also helps in packaging this lambda phage DNA inside the phage head. Now you would be knowing that we are talking about bacteriophage virus over here. So the lambda phage DNA has to be sent or has to be packed inside the head of the bacteriophage virus. So this COS site will be helping in packaging of the lambda phage DNA in the head. Apart from that, COS site provides a site from where the terminase enzyme will be cleaving, producing cohesive terminals, and different lambda phage DNAs can combine together and then they can be packaged inside the head of the bacteriophage virus. There are certain important points that I want you to understand before we move on to the Cosmid vector details. Now we are talking about the enzyme terminase over here. So let me just highlight this, that we are talking about the enzyme terminase. Okay, right. So enzyme terminase has two types of subunits. One is the larger subunit and one is the smaller subunit. However, we are not interested in going into the detail of the terminase enzyme. I just want you to quickly get an overview or a bird's eye view that what is terminase and how does it function. So what is the function of terminase? It cleaves DNA at COS site, as I have already told you, and helps in packaging the DNA into phage head. Next is, what is the target site of this terminase enzyme? The target site is COS N site. Understand this, that COS N site is present inside the COS region or COS site only. So COS N is a particular region. It is present within the 200 base pair COS sequence, as I have already told you. Next is use in cosmids. What is the purpose? Why we are using COS sites in cosmids? Because it will enable packaging of recombinant DNA into phage head and efficient transfer. Now you would be aiming at transferring the vector, Cosmid vector, inside a particular host, right? So first of all, your gene of interest should be packaged inside the phage head properly. So this will be aimed, or this will be achieved by the COS site. Next is, what is the gene location? So this terminase enzyme is encoded by a gene called as gene A. This is this basically codes for the larger subunits, and nu1, this codes for the smaller subunits. So there are two genes that are involved. Gene A coding for larger subunits, nu1 coding for smaller subunits. Now these two genes are present inside the lambda phage genome, right? So lambda phage DNA has these two genes. Now it is not encoded in the Cosmid vector. Now this is an important point that you should understand. We are not putting in these two genes, that means gene A or nu1 are not present inside the Cosmid vectors. So now you would think, then how come terminase enzyme is produced? If you are not putting the genes inside the Cosmid vector, how will the terminase enzyme be encoded, or how will the terminase enzyme be produced? Remember that the lambda phage DNA has these two genes. Lambda phage DNA has these two genes. So through the lambda phage DNA only, the terminase enzyme will be produced, and this terminase enzyme will be cleaving our Cosmid vector at the COS site. So Cosmid vector only has the COS site. The terminase enzyme and the gene that is coding for the terminase enzyme is present inside the lambda phage genome. From there the terminase enzyme is produced, and then it cleaves the COS site of the Cosmid vector. This you have to remember. No gene A or nu1 gene is encoded inside the Cosmid vector, right? Now restriction enzymes open the Cosmid vector. As I have already told you, you can make use of the restriction enzymes, and restriction enzyme will be cutting off the Cosmid vector, cutting of the Cosmid vector at unique sites. So there are unique sites present inside in the Cosmid vector. I told you EcoR1 site was present, or SMA1 site were present. So there can be different sites present on the Cosmid vector. Cosmid vector can be opened up. Now your gene of interest or your DNA insert, whichever DNA or whichever gene of interest you have, it should also be cleaved with the same restriction enzyme which you used for cleaving the Cosmid vector. Suppose you are cleaving the Cosmid vector with EcoR1 restriction enzyme, the same restriction enzyme should be used for cleaving your gene of interest. Why this? Because using the same restriction enzyme will ensure that you have the same sticky ends, and with this you can easily join your gene of interest inside the Cosmid vector. I would be showing you how this is done in just a moment. So after the DNA insert is gene of interest is cleaved off from wherever source you are taking the gene of interest, it is cleaved off from there, and then the Cosmid vector is also opened up. They are annealed together. Annealed means joined together. You can use DNA ligase for joining these two. Next, long concatamers are produced which are appropriate precursors for packaging in lambda particles. Now let us understand this term by term. What do you mean by long concatamers? Concatamers means which has repeating units, and because of these repeating units are attached end to end, right? So first of all, let us say this was the repeating unit, then again this repeating unit, then again this repeating unit. They are joined end to end. Now when I say repeating units, what do you mean by it? What is being repeated? Let us just talk about this. So these repeating units, for instance, let me just quickly show you a diagrammatic representation and then we can move forward. So I'm just zooming in, and so that we can understand this in a better way. Let me just check out if it is visible to you or not. Let me just quickly move to the left so that everything is clearly visible. Right. So I think I will have to zoom out a little bit. Right. So this would ensure that you are able to see. Yeah. So now let me make you understand the important features over here. So this over here is your Cosmid vector. Cosmid vector, right? So let us just discuss the important features of Cosmid vector. I believe all of you would be remembering that. So we said that the restriction enzyme or restriction site is present over here. Restriction site will be aiming at cleaving the Cosmid vector. Over here there is a cos site, there is a ori site over here, there is selectable marker over here. So all of these things we have already discussed, right? Now what would you do is you would be using a particular restriction enzyme that will be cleaving at the restriction site. Let us say that the cleavage is done over here. This is the restriction site, so the cleavage is done over here, and because you use the restriction enzyme, what happened is the Cosmid vector opened up. Previously it was circular in nature, now it has become linear. Okay, now you will use the same restriction enzyme. For example, I used EcoR1 for cutting the Cosmid vector. The same restriction enzyme would be cleaved in order to produce your gene of interest. So over here you can see partial digestion of high molecular weight DNA to give 30 to 40 kb. Remember that the gene of interest that you are using and the DNA insert that you want to insert over here, that should be roughly around 30 to 40 kb. That is why you are using partial digestion over here. Now there are two types of digestion. Let me make it very clear to you. Complete digestion simply means when you are using or when you are aiming at cutting at all the restriction sites simultaneously. That means if there are 3 restriction sites, I would be cutting at all the 3 restriction sites simultaneously. This is called as complete digestion. But when I am talking about partial digestion, it simply means that there is equal probability of cutting the 3 restriction sites. Suppose first restriction site can also be cut, second can also be cut, third can also be cut. There is a probability that none of the restriction sites will be cut, and there is a probability that all the 3 restriction sites can be cut. There is a probability that 2 of them will be cut, so on and so forth. So this is called as partial digestion. In case of partial digestion, you generally use lower concentration of the restriction enzyme, and then you can achieve partial digestion. So what is our aim? Our aim is that we want to code 30 to 40 kb of DNA in our cosmid vector. So what we have done is, we have, through the restriction enzyme cleavage of the genome, let us say that this was the genome of some organism, let us say this was the genome of some organism, and through eco R1 cutting, so I had cut over here, over here, over here, over here, so I would be getting different fragments of different lengths. So let us say that I am interested in only the fragments having 30 to 40 kb size. So what will be done over here in the next step, the restriction, the gene of interest would be attached like this. This is the gene of interest in between the two cosmid vectors. So let us say this is cosmid vector number 1, this is cosmid vector number 2, and in between your gene of interest is present, something like this. That is why I was referring to it as concatameric region, so long concatamers. So what is happening over here? So two cosmid vectors are present end to end, and one, let us say, gene of interest is present in the middle. And since this is also having the cos sites, this is one cos site and this is another cos site. I told you that cos site plays a very important role in packaging this DNA inside the lambda phage head. So how does it help? It tells the lambda phage machinery to package a particular DNA strand when there is the difference between the cos site of a particular length. That means, in other words, if I want to explain you is that if the length between the two cos sites is, let us say, x, then what will happen? The machinery of the lambda phage vector, it will be packaging this DNA inside the head of the lambda phage. Okay, so let us say that the distance between this cos site and the distance between this cos site is, let us say, x, and this distance will allow the packaging to be proper and efficient. So if the distance between the two cos site is at a particular level, then only the packaging will be done. I will be giving you the details in just a moment. So next, what will happen? Inside the lambda phage vector head, this DNA will be packaged. So let us say that the DNA is packaged over here, and then this lambda phage vector this lambda phage virus would be allowed to infect the E. coli or infect the bacteria. So now remember, you know that the cosmid is present over here. The cos vector or the cosmid vector is present now inside this cosmid vector. So cosmid vector is present inside the head of the lambda phage or the bacteriophage. Now this bacteriophage or the virus will be allowed to infect E. coli, and those E. coli that will be having the vector or cosmid vector will be resistant to, let us say, ampicillin, because in the vector you can see ampicillin resistance gene is present. So those E. coli which are having this vector would be ampicillin resistant, and those E. coli that are not having this ampicillin resistance gene will be killed in the presence of ampicillin. So I believe you would have got a clear understanding. This will also give us an impression that inside which of the bacteria our gene of interest is present and in which bacteria our gene of interest is absent. Now this is another diagrammatic representation. Basically it is giving the same information, just in a different way. So this is the source DNA, this is the cosmid. Now we have used a particular restriction enzyme over here, and because of the use of restriction enzyme we are getting the linear DNA, linear cosmid vector, and over here there is the cos site. So right, so then what you do is you also cleave with the BAM H1. So after cleaving with BAM H1, the cosmid vector is divided into two parts. So this is first part, this is the second part. So this over here was BAM H1, this over here was there was another this over here is BAM H1, and this over here is SCA1. So you are cleaving with both of these, that is why you are getting two fragments. Then what you do is you are cutting your gene of interest also with the same restriction enzyme. For example, SCA1 you cut, and then this is your gene of interest in between the two fragments like this. Then you allow the packaging because of the cos site, and then this cosmid vector or this lambda bacteriophage or bacteriophage will be infecting the E. coli. So this is what is happening over here. This is just the same thing, just represented in a different way. We are cleaving the cosmid vector by two enzymes over here. In this diagrammatic representation, we just cleaved it through one enzyme. Now let us move on to the next important thing. So we have discussed about annealing. Now so when you are packaging, or when the lambda bacteriophage decides to pack the DNA inside the head, what happens is it checks out that what is the difference, what is the distance between the two cos sites. So when the distance between the two cos sites is roughly 38 to 52, then this entire DNA is taken up and it is packaged inside the lambda phage head. Now fragments are produced using partial digestion. I have already told you the insert size or the gene of interest that can be used over here is roughly 40 kb long insert. Now of course this is very high if you are comparing it to a plasmid vector. Plasmid vector, if you are up to 20 kb, roughly 20 kb can be inserted inside a plasmid vector, but over here you are just doubling the insert size, infecting the host cells like lambda particles. Now this is a very useful feature of the cosmid vector. Why? Because you are packaging the cosmid vector inside the lambda bacteriophage, and this lambda bacteriophage has an intrinsic ability to infect the bacteria. So you do not have to put in extra efforts for transferring the gene of interest inside the bacteria. That will be done completely by the vector because it is having that intrinsic capacity. Next, inside the host, when this vector reaches inside the host, it replicates and propagates just like plasmid. Why? Because it is having the origin of replication, which will aid in their replication. I have already given you the diagrammatic representation. Now let us talk about the applications of cosmid vectors. Remember, genomic library of eukaryotes are prepared generally by using cosmid vectors. Why? Because eukaryotes have a large genome size. If you want to put in the inserts or DNA inserts inside a particular vector, the vector should have a high insert carrying capacity. So plasmid, as I've already said, it just has roughly, or at the highest level, it has 20 kb of insert size limit. But in case of cosmid, the limit just doubles. So obviously scientists want to use such a vector that has a higher insert size capacity. So for constructing the genomic library, large fragments are utilized, and therefore cosmid vectors are used. Since lambda particles are used to package them, many-fold more efficient than plasmid transformation. So when you are transferring your gene of interest inside the plasmid, and then the plasmid goes and inserts inside the host bacteria, this particular thing in which the lambda phage inserts inside the host is much more efficient, because lambda phage it has an intrinsic capacity to infect the E. coli cells or particular bacterial cells. Right. So now let us move on, since we have already discussed about the features of cosmid vectors and the efficiency of the transfer and the details with which the cos sites are helpful, terminase enzyme, etc. we have discussed. Now let us move on to the PYQs. Okay. So the first PYQ that I want to discuss with you is, which of the following statements about cosmid vector is true? Okay, remember we have to focus on the true statement. Cosmid contains only cos sites and lack any plasmid origin of replication. It has origin of replication, so this statement cannot be true. Cosmids are capable of autonomous replication like plasmids. So this is true, because just like plasmids, they have origin of replication. Cosmids can package insert size larger than 100 kb. No, this is false. Roughly around 40 kb can be inserted. Cosmids do not use lambda phage packaging system. This is also wrong. Cosmids actually use lambda phage packaging system, because you remember we have talked about the terminase enzyme, we have talked about the genes that are responsible. So yes, lambda phage packaging system is used. So the correct option is B. Let us now move on to the next question. The cos sites present in a cosmid vector are important because they acts as promoter of transcription. No, we have not studied anything like that. Help in packaging DNA into the phage heads. This is correct. Provide antibiotic resistance. No. The cos sites actually do not provide antibiotic resistance. Initiate DNA replication. Again, there is no role of cos site in DNA replication. Next. What is the approximate size range of DNA inserts that can be accommodated by cosmid vectors? It is roughly around 35 to 45 kb. We have already talked about this. Next, which one of the following is not a feature of cosmid vectors? Cos site for lambda phage, yes, this is a feature. Selectable marker gene, yes, this is also a feature. Plasmid origin of replication, yes, this is also a feature. Gene for capsid proteins, no, this is not a feature. So this is the correct option. We have already studied about the composition of the cosmid vector. There was no gene responsible for producing the capsid proteins. Now, in comparison to plasmid vectors, the major advantage of using cosmid is, why do you want to use cosmid vector? Higher transformation efficiency, this is correct. Ability to clone larger fragments of DNA, this is also correct. Lack of antibiotic resistance gene, no, this is wrong. Presence of multiple restriction site, no, this is also wrong. So we have already discussed that high transformation efficiency is present when you are using when you are using the phage vectors, because they are bacteriophages. They have intrinsic capacity to infect the bacteria. So obviously there is a high transformation efficiency. Apart from that, larger fragment or larger DNA insert size is also seen or can also be seen inside the cosmid. So correct option is A and B both. Let us move on to the next question. Cosmid vectors are generally used in construction of genomic libraries because they carry large fragments of DNA. We have already discussed this. So I hope you got a clear understanding about the cosmid vectors and the different aspects related to it. If you have understood the lecture properly, just hit the like button and subscribe to our channel, and you are free to tell us that which topics you want to study in the next lecture. Thank you so much. Have a good day.